Today, the topic of today's podcast is obsessive-compulsive disorder, OCD. And I have an expert with me from the UK, Cambridge, Professor Trevor Robbins, who's a professor of cognitive neuroscience and former department head of psychology at Cambridge. He's director of the University of Cambridge Behavioral and Clinical Sciences Neuroscience Institute, which he was involved in creating. He's a very well-known, widely published neuroscientist who expertise is in behavioral neuroscience, psychopharmacology, what neural circuits are involved in regulating, controlling specific behaviors, how drugs affect that, and then also in pathologies, and particularly psychiatric disorders that can result from dysregulation in those circuits. Trevor, welcome. I noticed I went to your wiki page, and I noticed you're a pretty darn good chess player. And I've been playing chess on chess.com, and I'm not, I can't get I have a low rating. It's like 1200, relatively low. But, the way the thing works, it's hard, it takes a long time to get
to ramp up. Yeah.
Yeah.
Yeah. Well, I used to be really good when I was young.
Yeah.
I played for the England juniors, and I was in the top 20 in the UK many, many years ago.
Oh, wow.
But nowadays, as you get older, the working memory declines a bit.
Yeah.
And it takes you longer to think, you get into time trouble, and you don't do as well. But I'm still quite a strong player, but nowhere near as good as I was when I was younger.
Do you play on chess.com?
No, I don't do that. I play over the board normally.
Yeah.
That's a fun website, I know. And I often follow tournaments in like the World Championship.
Oh, yeah. Because I was thinking maybe I can play you and see how badly you can beat me.
Well, one day we can certainly have a go at that.
Okay, so let's go. Why don't you go back to your the early years? So you've spent pretty much all your time at Cambridge.
Yeah, I'm afraid so. My excuse is every time I've been about to leave, I've been promoted.
Well, they love you. They love you there. That's why.
Kind of. Yeah. Anyway, I've had some opportunities to leave, and it's never quite worked out. But I found it's a great base, to travel a lot. And so I've traveled to the States over 250 times, I think, and all around the world, to lecture and visit labs. So I don't regard myself as being stuck in the mud,
Yeah.
It's just the way it panned out for me. I've been able to stay in Cambridge, and it's quite a good spot, and there we are.
I got a long time ago. So you were at Jesus College, right? So I think, people in the United States might not, say college. We think of university, but.
Yeah. Well, Cambridge University and Oxford University are what are called collegiate universities, and they comprise a kind of federation of colleges. And Cambridge University has 31 of these. It's been added to over the years, obviously. There are quite recent ones. Jesus College is one of the older ones. It was like founded in the 16th century, actually. But I'm currently a fellow at Downing College, which was founded in 1800. And that they call that college the oldest of the new and the youngest of the old, because it's right in the middle,
I know, I have so when our son was 2 years old, this is a long time ago, Michael Safroniev invited me to come and give a talk at Cambridge, and it was during the summer, and so the students were mostly gone, and so he put us up in the infirmary at Jesus College.
Ah.
And so our son, he's 2 years old. He's running all around the different rooms in the infirmary.
Yeah. Jesus College is a good one because they have the sports playing fields there. So it's in a great green expanse.
Yeah, there were people playing cricket out the window.
Exactly.
Yeah. Okay, so you start out at Cambridge so your work started out in animal studies.
Animal studies, yeah. So my thesis was on the behavioral and neural effects of amphetamine in animals, in rats. And I was particularly interested. There is a theme which kind of runs through my work. So I got really interested in stereotype behavior
Okay.
produced by amphetamine. And, you know, Susan Iverson and Ian Crease discovered that that was due to dopamine in the caudate nucleus and the putamen in rats. Whereas locomotor activity produced by amphetamine at lower doses was present in the, you know, is mediated by the nucleus accumbens and the mesolimbic system. And that was quite a big breakthrough in the mid-70s. And I traveled over to Denmark to work with somebody who unfortunately is dead now, Melvin Lyon, working with Randrup and Munkvad, who were some of the first people who proposed that dopamine was involved in schizophrenia, actually.
Yeah.
And I did some studies with them, but also I did the big review with Melvin Lyon on the behavioral effects of amphetamine, and we had a theory about it, which included some neural substantiation as well. So then I kind of built on that, you know, really got interested in other transmitters other than dopamine, got interested in the frontal cortex as well as the striatum, got interested in humans and non-human primates as well as rodents, got interested in Alzheimer's and Parkinson's disease first, because Parkinson's disease is an obvious dopamine syndrome, although as you know full well, it's much more than just being a loss of dopamine these days.
Yeah.
And then we moved on into, we invented this neuropsych battery called Cantab, which was the first, I think, one of the first computerized test batteries using a touch-sensitive screen. And it was based on animal paradigms, you know, translated to humans and vice versa. We tried to do a bit of both. So we converted the Wisconsin card sort test to an animal learning test, which we also gave to humans. It turned out quite sensitive. And so we're measuring things like cognitive flexibility still, and then into psychiatry, and then, you know, you get into addiction. And ultimately, I got interested in compulsive behavior, which is, again, perseveration, like stereotypy originally.
Yeah.
And then the ultimate compulsive behavior, as we know, is in OCD, obsessive-compulsive disorder. So I put a lot of my activity into studying OCD in recent years, and if you like, animal models or relevant animal preparations to understand OCD.
So yeah, with your very first work in amphetamines, so you mentioned the basal ganglia, the caudate and putamen or striatum, as in the animals we call it, and this is kind of generally accepted, this brain region, to be involved in like habitual behaviors or almost unconscious.
Absolutely.
Repeating, right?
Absolutely. So this important distinction between goal-directed behavior, which is kind of conscious and executive and frontal lobe-like, you know, your voluntary behaviors, your goals of your desires, versus the more automatic stimulus response habit learning thing, which interacts with goal-directed behavior. You know, both of these systems are probably working at the same time to generate behavior, but there's always this not only a cooperation between them, but also a competition between them. And some syndromes, like, for example, drug addiction, Barry Everett and I suggested involves a bias towards the habit system, you know, which is consistent with compulsive drug seeking, for example. And similarly in OCD, you know, a bias towards compulsive behaviors which are mediated by dysregulated habits.
And it makes sense, even from an evolutionary perspective, right? That, okay, this is a simple thing, like when we're walking around town, we're not thinking about, you know, every step we're taking, or, you know, turning our head or this, you know, and, you know, so those things are going on, like these motor programs are, that our behaviors, actual movements are going on while we're thinking.
Yeah, you know, we have these long sequences, right, don't we, of behaviors. and a lot of them are fairly automatic. But we have to stop every now and again and just check that we're, on track to getting our goal.
Yeah.
Right. And we also have routines, like in the morning when we get up and, wash and brush our teeth and so forth.
Yeah.
And of course, these are pretty automatic things. And it's good that they're automatic, so that gives us some space to listen to the radio and to think about what we're going to do later in the day.
Right. Think about your next experiment in the lab.
Exactly.
Even while driving cars, I remember, we live north of Baltimore, and the NIA lab was in Baltimore, and so it's 30-mile drive. And a lot of times I get to the lab. I mean, I don't remember really any details of anything on the way.
Yeah.
Right. But you get there okay.
Exactly. Sometimes, the classic thing, isn't it? Sometimes you get in your car, you're thinking about something else, and you find yourself on the wrong road. You're going to the station rather than to the lab or something. So you get into another habit by mistake. It's called slips of action, and that's when our executive system isn't quite monitoring what we're doing properly.
Do you want to? So you mentioned this. Was it Cantab battery of
Yes.
neuropsychological tests? Can you kind of define what cognitive control is and what executive function is?
Yeah, absolutely. I've had the pleasure recently of doing this review with Naomi Friedman.
Yes.
Who's a fantastic expert on the psychometrics of frontal lobe assessment, as it were. And their classic theory with Miyake, who I've never met actually, I'd love to meet him sometime. But their classic theory is, postulates a triad of executive operations. It's like molecular psychology, this. Let's see if we can chop up executive function into its constituents. And their triad, which I like and I agree with a lot, is updating working memory. So we all know what working memory is. The way I like to talk about working memory to an audience is I say, so repeat after me the following numbers, 365878. And they all do that very easily. I say, okay, that's not working memory. Working memory is repeat after me 365878 backwards. That's working memory. Then there's response inhibition, which is inhibiting your impulses and your compulsions, probably. That's an interesting issue, behavioral inhibition. And thirdly, cognitive flexibility, our ability to multitask, essentially, and to switch our strategies and behaviors when appropriate. And, they tested this very objectively by giving a whole battery of tests with 3 tests in each of those categories, and showing that overall, all of the tests in an individual did agree in terms of performance levels. But nevertheless, there were clusters in these 3, these triadic components of executive function. So they support this idea that there is something called cognitive control or executive function. I think those two are almost synonymous, to be honest.
Yeah.
There is something in common with these things, but there are also specific executive functions as well on top of that. And, Naomi produced this really interesting paper showing that IQ was most related actually to working memory and not to cognitive flexibility. And that got us very interestingly into a big project in Singapore, Science of Learning, because of the interest in training kids and adults to be more creative in their thinking. So instead of training working memory, which obviously helps IQ, train cognitive flexibility, which may help your creative thinking. So I think that's an interesting line of work. So I'm very interested. Now, we don't think that those are necessarily the only components of executive function. There may be others, for example, generation, verbal fluency, when you have to say as many words as possible, beginning with the letter A, or as many animals as you can think of. Verbal fluency, generation is one. Monitoring performance is another important one. And Sylvia Bunge at Berkeley has got this very interesting idea about relational learning is one, higher order reasoning, analogical reasoning, that type of thing. So there may be other elements of executive function which we haven't really fully explored yet and understood in terms of whether they're separate or whether they're just part of this general executive function multiple demand system.
And what are so let's start talking about OCD now, I guess.
So OCD, we think of as being first of all an imbalance towards the habit system,
right,
and secondly dysregulation of those. So a loss of executive control may be a loss of inhibition, but maybe also cognitive inflexibility, an inability to shift between, for example, goal-directed and habitual behaviors, which some people call an arbitration deficit. And I think that kind of fits what we know of some of the neural basis of OCD, based largely on imaging studies in humans, whether structural or functional or resting state or whatever. The idea that there is impairment, for example, in connectivity between the lateral frontal cortex and the stratum, the caudatum putamen, and that may underpin problems in cognitive flexibility and also in planning. So OCD patients aren't terribly good at planning, which is obviously a form of goal-directed behavior, which depends on working memory and these other functions. So, I think that's one aspect of it. Orbitofrontal cortex laterally is also pretty inactive. And so there are impairments in reversal learning, which is another form of cognitive inflexibility in OCD. And several investigators have shown that. And because we've shown in non-human primates that depleting serotonin from the lateral orbitofrontal cortex impairs reversal learning. And we've just got a paper coming out actually in biological psychiatry, GOS, showing that in OCD, unmedicated patients are bad at reversal learning, and the medicated ones on serotonin selective reuptake inhibitors, which is the standard treatment, they do a bit better, even though, symptomatically, they may even be worse than the early in the course unmedicated ones. So there is something about, serotonergic modulation of the OFC maybe in OCD that's important. The other structures are the head ganglia, as it were, of the habit system, in the cortex, the supplementary motor area, and the anterior cingulate cortex. And we've just published a big paper on with GABA and glutamate measures of the SMA and the anterior cingulate, showing that in OCD, first of all, their clinical symptoms of OCD are related to the level of glutamate in the supplementary motor area. Secondly, a questionnaire about obsessive-compulsive tendencies, which you can give even to healthy people, obviously sky high in OCD patients. That's also related to increased glutamate. And it's even related in healthy individuals, although obviously at a much lower level. And then finally, we've got this measure of habit, which is a bit complicated. I can describe it if you like, which we've also used in actually non-human primates in marmosets with Angela Roberts, showing that the measure of habit is also correlated with glutamate in the SMA and also in OCD patients in the anterior cingulate. The glutamate GABA ratio, the higher the ratio, the more habitual in the humans. And that seems to chime in with these data we've got in marmoset monkeys when we manipulate the anterior cingulate.
So when someone comes to the clinic, so the diagnosis is based on like self-reporting and maybe a spouse, but then which specific tests are kind of the key ones that?
Oh yeah, okay. Well, of course, psychiatric diagnosis is not done with these tests.
Right. Okay.
That's what we have to realize. I mean, it's done by an experienced clinician, and I'm certainly not that. I'm not a psychiatrist.
Okay.
So they use something called the Y-box scale. and that stands for the Yale obsessive-compulsive scale.
Huh.
It was invented at Yale,
Yeah.
I think by Lechman, some Lechman, someone like that. And that's used universally. And it's a whole lot of questions that the clinician, goes through with the patient to establish that they have the criteria for OCD, which includes intrusive thoughts, obsessive thoughts, which go on and on in their minds. They're kind of ruminations, really. Intrusive, often unpleasant, and compulsive behaviors, which are repeated behaviors. Now, there's an interesting issue here semantically, because there's also this category called mental acts. So, if you think that if you count, for example, that's a kind of mental act, and that counts almost more as a compulsion than as an obsession, if you see what I mean. If you're thinking of doing something, that's a mental act.
Right.
And I think mental acts are related to compulsions. And I think, the reason that I focused on compulsions, of course, is because we can't really easily ask animals about obsessions.
Right.
But we can ask them about compulsions, as we did many years ago when we were looking at stereotypies in, produced by amphetamine in experimental rats. So compulsions are kind of easier to measure in a sense, and we can theorize about them. But I do think probably compulsions and obsessions are related in some way. I've actually, so the common idea is that, well, you have anxiety, obsessions, you have these anxieties, and to reduce the anxiety, you do this behavior, this compulsion, which kind of reduces your stress, reduces your anxiety. And I think there is something in that idea. It's like a negative reinforcement notion of compulsions. There's obviously something in that idea, but, sometimes we think that it may work a different way. It may work, instead of OCD, it may work COD. In other words, you produce these behaviors for reasons you're not really very clear about. You can't help yourself. And so you begin to invent reasons in your own mind for why you're doing those behaviors.
Yeah.
So, maybe you find yourself washing a lot, and sometimes that makes you think, well, maybe, why am I washing? I'm scared of germs, And untying that Gordian knot of causality and behavior and introspection is very difficult.
Well, there's an aspect of doubt, self-doubt, and did I get enough bacteria or germs off my hands when I washed them the first time? Maybe I didn't get enough.
Well, that's a very key thing about OCD, the just getting it just right thing. There's this idea that we've shown that they have elevated prediction errors in learning. So you obviously know what prediction errors are when your expected outcome there's a much bigger deficit when you actually get the outcome against your expected outcome. And obviously, if OCD patients have elevated prediction errors, which we've shown and others have shown, like Tobias Hauser, they're going to repeat the behavior until they reduce that error. And as we all know, the anterior cingulate is very importantly involved in error monitoring. And there is this potential called the error-related negativity, which probably emanates from that region and is elevated in OCD. So, this is again part of this circuit, which I think.
Now, we've talked about, what about, it's kind of the classic way in animals that you try to understand is this specific brain region, or even these specific neurons involved in a behavior. You can actually kill those neurons.
Absolutely. And that's exactly why we do animal experiments, right? Because we're interested in causality. Because it's so darn difficult to infer causality from correlations with imaging, for example, or even with electrophysiology. Obviously, if your electrophysiological signal precedes the behavior reliably, you can be sure that the behavior isn't causing the signal. And so you infer that maybe it's causing the behavior. So those are the kind of things that we can do, obviously, more readily in experimental animals and humans. And lesions, classical lesions, and now the modern day opto or chemogenetic inhibition, obviously test that. And the thing which strikes me, Mark, is that, our search for treatments is really important because it's the ultimate test of the hypothesis, isn't it?
Yeah.
Now, if you've got a hypothesis that this is going wrong and you've got a treatment, a drug, for example, we're interested at the moment in mGluR2 receptor agonists.
So these are the particular type of glutamate receptor.
Yeah, to reduce glutamate release, to maybe normalize glutamate release in certain regions of the cortex. Maybe this would work. But if you've got a drug which actually works in the disorder, that's almost the best way of testing your causal hypothesis, isn't it? And of course, that's the big one, because as we know in psychiatry, that's incredibly difficult to do.
Well, Trevor, since I retired, I've written a couple books, but this one's going to come out pretty soon.
I should have a look at this.
Yeah, it's
okay. That's great.
Sculptor and Destroyer: Tales of Glutamate, the brain's most important neurotransmitter.
Oh,
and
God.
I try at one point in the book, I try to make the case that because if 90% of the neurons use glutamate as their neurotransmitter, and that's the main circuitry throughout all these brain regions you're talking about.
In the cortex.
Why aren't there a lot of drugs that, and I think that the reason is it's exactly because glutamate is so important. It's hard to get a drug that doesn't have really bad side effects.
Yeah, I know.
I think but the mGluR maybe.
Well,
it has more subtle effects than the.
Well, the mGluR2 is an interesting receptor because, as I say, it's a presynaptic inhibitory receptor on glutamate, and there is a drug. Do you remember pomaglutamide?
No.
It was a drug that Lilly invented and published in Nature Medicine in the early noughties, I think 2007 or 8. And they tested it against olanzapine, classic neuroleptic, and showed that this pomaglutamide, which was an mGluR2 and 3 agonist, I think, did as well as olanzapine and had fewer side effects. However, the problem was they couldn't replicate the study. And they dropped the drug, which I think may have been premature, because someone called Bruce Kinnan published a really interesting paper in Biological Psychiatry many years later, I think 2015, showing when you did a reanalysis of the data, that the pomaglutamide drug did work in those patients who were relatively early in their symptoms. It's first episode patients. Worked for a couple of years, as it were. And it struck me that may have been because in early schizophrenia, there's this surge of activity, hippocampus and probably also the cortex, which must involve glutamate. And then probably it undershoots a bit. So, it may lose efficacy. But if you had a drug which blocked the initial psychotic breaks, that would be cool, because they actually produce the chronic schizophrenia. Something I've chatted to Danny Weinberger about in the past.
Well, but you may have like an early major structural change. That then is after that is in place. It's it's hard to affect.
But you're absolutely right. I mean, so we've got this recent paper on glutamate and OCD. So we showed really significant increases in the anterior cingulate and significant reductions in GABA in the same patients. So, we're very excited about these data. But of course, there is the point that probably in other areas, glutamate is normal. So if you give a drug which reduces glutamate globally, what is that going to do? We'd like to, reduce that elevation if it's causally related to symptoms, which it may or may not be. So, that's the classic problem, isn't it, with systemic drugs? And I just wonder in the future whether we're going to have to do more neurocircuit-based manipulations,
Yeah.
TMS, and so we've done this study with deep brain stimulation in OCD, because actually, OCD is a great model, I think, for treatments in all modalities. So first of all, CBT with response prevention is reasonably effective in OCD. So there's a behavioral treatment. Right. We're interested in something called habit reversal in OCD, which I'll describe later if you like. Then SSRIs kind of work a bit. Nobody really quite knows why or how. We think it's because of this frontal cortex thing.
Or it could be, you mentioned anxiety.
Could be that. We're not, I don't think anyone's quite sure what SSRIs do in OCD. They're used a lot and they seem to be quite effective. Is it treating the emotional problems, or is it treating the compulsions, or is it treating both? Not quite sure.
Yeah.
And then, there's newer drugs like glutamate agents, for example, or maybe even GABA agents. And then you've got circuit treatments like deep brain stimulation and TMS. I mean, Rob Malenka tells me that in Stanford, there's some very interesting work going on with TMS at the moment. And obviously, the only form of psychosurgery that I can think of that's ever shown any evidence of efficacy and benefit to the patient, very severely affected patients, is cingulotomy.
Huh.
Some neurosurgeons are still doing cingulotomies in these patients who are so incredibly impaired with OCD, they can't leave their houses and they're performing rituals 12 hours a day.
So they're removing cingulate cortex tissue?
Yeah.
But isn't that...
It works.
Isn't this... Okay, that's interesting. Huh.
It's amazing.
Yeah.
And the neurosurgeons I talk to, some of them in the UK, prefer to do that than to use DBS, deep brain stimulation, which one might have thought is a more refined treatment.
And they have to remove on both sides?
They do, I think. Yeah, they do. It's kind of a midline surgery, of course, on both sides.
Just for those who don't know, most of these structures are bilateral that we're talking about.
But I think if you Google it, you'll find that there's a lot of supportive evidence that the anterior cingulate is involved. There's a very influential paper in Journal of Neurosurgery. I'll have to check the authors for you, but it's quite recent and it's very, very persuasive, actually. Huh. In our study of deep brain stimulation, the only one that's been done in the UK on 6 really impaired patients. This is in collaboration with the neurosurgeons at Queen Square, the National Hospital, and Professor Eileen Joyce, who's the professor in neuropsychiatry there. So we had 6 really severe patients. So their average Y-book scores, Mark, were 36 out of 40.
Okay. Wow.
So that equates to not leaving your house and spending an awful lot of your time doing your compulsions and obsessions. So these people signed up voluntarily. No treatments have worked. And, this is one of the criteria. They are completely treatment resistant. So they get electrodes implanted. And what they did was they compared 2 sites in the same individuals, which are 2 sites which have previously been shown to have some efficacy in different studies. So the French group, for example, has shown that electrodes in the STN, our favorite subthalamic nucleus, as in Parkinson's, they've claimed that works. Damien Denise and others, and Stephen Rasmussen and lots of others in the States have studied effects on ventral capsule placements, which are close to the nucleus accumbens in the internal capsule, in the middle of the stratum, as it were. That's supposed to be effective. So in this very small study, we compared the 2 sites in the same people, stimulating for 3 months, first one site, then the other site in counterbalanced way, and then both sites together. And then they added CBT at the end, just to be sure.
So the subthalamic nucleus is involved in control of motor behavior, body movement. So is there? So what are these people? Their perception of that they're not repeating the act, but Is there a psychological aspect?
Let me tell you a bit about it. So first of all, the STN, I'm not a world expert, by the way, on the STN, but the STN has different compartments.
Okay.
And some parts of it are motor, but some parts of it are not motor and are probably involved in cognition and emotion. And we put the neurosurgeons put the electrodes in the anterior medial part of the STN, which is the limbic emotional part of the STN. Okay, so they compared these two sites, and guess what? 5 out of 6 patients, it's never 6 out of 6, of course, but 5 out of 6 patients showed fantastic improvements. And when you take the average, even of all 6, the scores were reduced by around 40%, I recall. So going from 36 way down, to 10 or something like that, something amazing. And
what's a person who doesn't have any obvious obsessive compulsive behaviors? Where do they score on that?
Well, I think the technical threshold is about 8.
8, okay.
So if you get below
this stimulation is getting them down up pretty much to normal.
Getting down almost to normal in 1 or 2 cases. And people going on holiday to Barcelona, things like that, not having left their homes for months. So that was amazing. Now, one of the patients didn't respond very well, but 5 patients did respond well to both sites. And overall, both sites worked the same in terms of quantification on white box. However, the really, really interesting thing is when they took other measures, supplementary measures, it was clear that the ventral capsule stimulation mainly affected mood, anxiety and depression. And we think goal-directed behavior. Okay, whereas it didn't improve cognitive flexibility. Whereas the STN stimulation mainly affected cognitive flexibility and didn't improve mood.
Wow.
So then they did tractography and showed that the 2 sites actually stimulate different pathways. So the ventral capsule stimulation is probably stimulating largely medial, orbital, and anterior cingulate, actually, regions, whereas the lateral frontal cortex is mainly affected by the STN stimulation. And that fitted with, our evidence from non-human primates that, and humans indeed, that those structures are more involved in cognitive flexibility. So I was really excited.
And do they, when they're placing these electrodes, they use CT scan, or what are they using to?
Oh, they have incredible MRI. They have incredible accuracy in placement. These guys, they're like, precision engineers.
And it doesn't matter that they're using the electrodes are metal in the magnet because they're so small, or
oh yeah, I mean,
or maybe there's some other conducting metal that
they use some other. I don't know the technical details.
Titanium, or I don't know.
But they are incredibly. And by the way, they don't. In our group, they didn't think that the ventral capsule electrode was affecting the nucleus accumbens. They think it's affecting fibers of passage going to the anterior cingulate. And I think that's consistent with the type of ideas that Suzanne Haber, for example, who's now at Harvard, has, and Tony Grace, who's worked with animal models. They've both suggested that kind of placement actually affects cortical structures. But what I found exciting about our study and original about it was the idea that you've got maybe different circuitries contributing to different aspects of the disorder.
Yeah.
And, that has implications for treatment, obviously. And whether you could get TMS working to selectively affect these different circuitries, that would be, then you could begin to treat patients who aren't very, very severe, because obviously you can't.
But with the electrodes, they're implanted and they have a battery thing in there. Yeah. But with the TMS, you have to go. I mean, you can't have a TMS, walking around.
No, you just have to administer it, don't you? And then it lasts for so long, I suppose.
Yeah.
I don't know the exact protocols. I've never used TMS, but I'm told that there are obviously published reports that TMS, repetitive TMS, is doing quite a good job in OCD.
Yeah, interesting.
Mm.
And
So
what about?
And I think the other exciting prospect for the future, and this is a bit science fictiony, but maybe dreads will work eventually in humans.
Oh,
So
now you're talking about a method where so the idea would be you would use probably a viral vector to express an ion channel in the neuron, and then that is engineered so that a drug will activate the ion channel or inhibit, depending on what you want to do.
Absolutely.
And so you'd inject. There's
So you have a
viruses.
surgery.
One's called AAV, right? Adeno-associated virus.
You have one surgery to get your AAV in, or whatever it is.
And you put it only in the brain region where you want.
Yeah, exactly.
Yeah.
I reckon it'll take about 30 to 50 years before we're able to do that in humans.
But has that, okay, so you mentioned animals, right? And so what about animal models? Obviously, that's something that can be done.
This idea comes from, the brilliant work of Suzanne Amari and Eric Bergier in Paris, where they did this great set of studies with optogenetics, where Suzanne's group activated medial orbital frontal cortex in the SAPAT mouse.
Okay.
And produced essentially, it affected the striatum, but it produced effectively compulsive grooming, which was their kind of animal model of...
By stimulating the frontal cortex.
Yeah, by stimulating the medial orbital, because in OCD, the medial systems are more active.
Oh, okay.
And the lateral systems are less active.
Okay.
The lateral systems are less active.
I see.
What Eric Bergier did, just supporting that idea, is that he stimulated the lateral orbital and was able to inhibit. So there's a kind of yin and yang between the lateral and medial orbital frontal cortex in OCD, maybe as well, but certainly in these animal models. So he got compulsive grooming in one way and Suzanne in another way.
Someone could try the genetic engineering essentially thing with, you talked about amphetamine causing stereotypical behaviors. I mean, you could try that, and that's in some sense similar to OCD.
Well, it is. Yeah, I think it's a very simple. I would think of, well, to be honest with you, I think of the grooming, and I think Suzanne may agree with me, I'm not sure on this, but I think the grooming is probably a better measure of what we call trichotillomania, which is, repetitive hair pulling, which may be a primitive form of OCD. Okay. So that's what and maybe even Tourette syndrome, which is ticky. Maybe that is a bit like
you know what, Trevor, this is very interesting. So my father and my son, actually, I don't do it, but when they're thinking very, deep thought, they twirl their hair. And I chew my fingernails. And somehow that, I don't know whether it's like a background, like
maybe it helps you focus.
Yeah, it helps me focus, but I'm not thinking as much as I used to. My nails are a little longer now than they used to be before.
But the other interesting thing is, look at Rafa Nadal, the tennis player, when he's serving. Look at the ritual he undertakes for each serve.
Yeah.
He's using this ritual to prepare his motor system to do the best possible serve.
Yeah.
And so these rituals do have some adaptive function.
Yeah.
you know, they reduce anxiety a bit,
Yes.
probably,
Yes.
maybe disruptive as well.
Yes.
So one idea, of course, is, as I was saying earlier, that compulsions are, you know, regulating anxiety.
Yeah.
And I think, but what I really think is that may be the case early on in OCD, but it's a bit like avoidance behavior. You know, avoidance behavior may be driven by anxiety initially, but then it may become an automatic behavior.
Yeah.
So there's quite a lot of evidence that avoidance can become habitual very quickly.
Yeah.
And so the anxiety then is less important in maintaining the behavior than in initially initiating it and generating it.
Yeah.
So that's my theory about compulsion.
Now, there's some genetic factors that are involved in some psychological disorders. And, you know, previous podcasts, we've talked about this in relation to all sorts of different disorders, schizophrenia, Alzheimer's.
Alzheimer's,
Well, what's the situation with OCD?
The situation in OCD is not so clear.
Okay.
I don't think, now I'm not the one to ask about this really, but my understanding of the latest GWAS is that there is no really very, very, very strong candidate for OCD. You know, studies have thrown up several genes which, you know, may be relevant. So I mentioned one, the SAPAT gene, which Susan Amari's work was based on in the mouse. I think Feng from the Broad Institute was involved in generating the transgenic mouse on this, which is a very clever thing to do. And SAPAT protein is a structural protein associated with the AMPA receptor, and it affects straight or glutamate transmission. So, you know, it sounds like a good candidate for OCD. And there's another gene called SLIC, I think, SLIC, and I can't remember the number, which is implicated in glutamate reuptake. So I like this, you know, this is a glutamate story.
Yeah.
But I don't think any of these genes have yet attained, you know, GWAS acclaim, as it were, in the same sense as they have in schizophrenia, for example. So obviously, I mean, it seems pretty clear to me that it's obvious in families of OCD patients that their relatives also have these tendencies. Even, you know, they don't have OCD diagnosed, but they have the tendencies. So, for example, we've shown that they're less flexible. They tend to be, you know, less able to inhibit, and there may be other, you know, problems. Also, they have less activity of the lateral orbitofrontal cortex during reversal, and we published that in Science with Sam Chamberlain and Barbara Sahaki in around 2008 or 9. So, you know, there are these endophenotypes which suggest that there may be some genetic predisposition, but, you know, actually nailing it down and identifying the real genes involved, I think we're not quite there yet with OCD. We need some major studies to do that, I think.
And what about age of onset of, you know?
Oh, that's fascinating. Oh, Mark, that's fascinating, because it can come on really early. You know, and you probably, I don't know whether your own kids, you can see it even in your own children's, very early on, age 4 or so, they get into little stereotypies, you know, during play, and they generally grow out of that quite quickly, but some of them don't. We found we did an analysis with some colleagues from China, actually, very clever at data analysis. We analysed a lot of data from the so-called ABCD project, which is, you know, this brilliant longitudinal project that NIH have commissioned with Terry Jernigan and Sandra Brown and others, looking at every aspect of children development, including genetics, imaging, behaviour, you know, the whole thing, from age 10 onwards. And we identified two types of kid. One kid involved in a lot of repeated behaviours, and this was associated with less grey matter in the frontoparietal system, I think, which is what we'd predict, the executive system. And another group who were more, they had problems with kind of body image and perfectionism, but we couldn't see any obvious neural correlate of that. But, you know, so these tendencies, I think, are there pretty early on, actually. and of course may develop into OCD. And I think the tragedy of OCD is because it's often trivialized stupidly as, not being, we're all a bit OCD, OCD is not really a proper mental health disorder. It can be really serious. And the longer you leave it untreated, the worse it will get. In our terms, because you're building up those habits. And, once you build up habits, they're very hard to move.
Yeah.
That's our theory. So we think you should be treating OCD very early on. We're currently focused a lot on adolescent OCD.
Yeah.
But that, I think that's a real big issue for the future.
And people with kids with autism one of the diagnostics is repetitive behaviors, right?
Yeah.
And I don't know if there's but what about the early life environment? Have there been any evidence of any?
It's hard to say. It's, the literature just isn't there. I mean, one assumes that these predispositions are at least partly genetic, but obviously there may be environmental trauma, maybe even some stress that happens early on, which, scares the life out of you and makes you potentially, habitually prone or whatever.
Yeah.
It's a really important area, but so poorly understood. And of course, Judith Rappaport did some great study in IMH on this early on with Swedo. And there's one paper in particular which we've cited in this review we did in Neuron, where they describe, talking to these young kids, and these kids don't really have any clear idea why they're doing the repetitive behaviors. It doesn't seem obviously to be due to anxiety or anything like that. So I think the call is out. On the other hand, you could say that young kids aren't very good at understanding their inner motivations. Do you know what I mean? They may even not know quite what anxiety means. So then it becomes a language semantic problem of how you interpret their behaviors.
So in most instances, the problems begin relatively early.
I think so, yeah. I mean, they talk about adult onset OCD and child onset OCD, and I think that is an important comparison we have to make. But I suspect my clinician friends tell me that many cases of adult OCD started much earlier.
Yeah. Now, one aspect of this is that, so the prefrontal cortex, there's this literature that it develops more slowly than other brain regions, at least in terms of myelination and
absolutely
connectivity.
Yeah. I think we all sign up to that one. Yeah.
Yeah. And certainly kids, adolescents for sure, they're more impulsive just in general. And so the idea is maybe, this late development.
So this is the interesting thing. Remember earlier we were talking about behavioral inhibition.
Yeah.
Right. Which may be impaired in OCD. Obviously, it's probably also impaired in ADHD, attention deficit hyperactivity disorder, which is, really a different kettle of fish. This is, kids who are distracted and they have inattention and they're impulsive. So I've been very interested as well in this related construct, which is much more studied than compulsivity, of impulsivity. And so impulsivity, I think of as behavior which classically is prematurely expressed,
right,
risky, often has bad outcomes. Not always, it can be good, but often has bad outcomes. Compulsivity is behavior which persists despite adverse consequences, So that's the difference. Now, in both cases, one imagines that behavioral inhibition, inverted commas, must be playing a role, but presumably in different circuits. And, Jeff Daly and I have written about this in our review, Nature Reviews Neuroscience, that we think that there are different circuits for impulsivity versus compulsivity. And this goes back to the theory that Barry Everett and I had on drug addiction, which is we think that impulsivity is a vulnerability factor for many forms of drug abuse, particularly stimulants. And that involves changes in the nucleus accumbens and structures like the infralimbic cortex and in the rat and the prelimbic cortex. Whereas compulsivity is much more to do with the caudate nucleus and putamen and the orbitofrontal cortex, right? So that's their argument, that impulsivity and compulsivity are both disorders of behavioral inhibition, but they involve different circuits.
Yeah, interesting.
Yeah, and you know, this idea of inhibition also extends to structures like the amygdala, emotional regulation, to the hippocampus, memory regulation. You know, Michael Anderson stuff on forgetting, directed forgetting, you know, involving inhibition of the hippocampus from prefrontal cortex. So I think inhibition may be the general executive factor, I think, in understanding frontal cortex.
So does this fit? You know, we can finish up pretty soon, but
oh, really?
One of your
enjoy this.
Okay, one of the papers that I read of yours, I think it was a review. You talk about the idea of unity and diversity.
Yeah, that's with Naomi Friedman.
Yeah, and
paper in Neuropsychopharm, which came out last year. So, yeah, I really just collaborated with Naomi to review this area. You know, the idea that that there is something core, multiple demand, called the executive function, which all tests of frontal lobe function use. But diversity means that there are also specific aspects of executive function which are separate and involve things like inhibition, cognitive flex, and working memory.
Yeah, but there's circuits in many different brain regions are working together all the time, and so
the way I would think about it is that there are hubs, right, in the frontal cortex. We get that from small world analysis by people like Ed Bullmore at Cambridge. You know, there are hubs which are incredibly hyper-connected, and they can subserve the kind of multiple demand system things that John Duncan and others propose, you know, common executive functions. So in other words, in theory, the same neurons are doing very different functions in different contexts. So they're super neurons, super ensembles. But each of these hubs may have rather specific circuitry associated with them, which subserve different elements of executive function, like flexibility, like switching, like inhibition, and holding online. Particularly this lateral frontal parietal system, which everyone highlights now, being involved in cognitive control.
Yeah, and my most recent podcast with Rob Malenka, we spent some time talking about the fact that, you know, throughout the cerebral cortex, these circuits they're excitatory glutamatergic neurons, inhibitory GABA, and then you have these axons coming from neurons use dopamine or serotonin, and those neurons are few in number, and they're located in kind of more primitive brain regions, you know, brainstem, and somehow, which I don't think anybody's figured out, somehow they seem to be modulating what seem to be fairly specific behaviors in important ways.
Yeah, now this is one of my most important research questions that I've pursued the whole my life. You know, this is the bit which puts together my psychopharmacology with my cognitive neuroscience. So the big issue is, what are all these systems doing that's different? Dopamine, noradrenaline, acetylcholine, serotonin, histamine. They're all coming from the brainstem and midbrain. They're innervating probably subtly different regions of frontal cortex, and they're all involved in stress.
Yeah.
So what is it they're doing that's different? They can't all be doing the same thing. So they must be modulating different parameters of these processes. And, you know, we have some clues on this. So our biggest clue has been serotonin and reversal learning. So serotonin, you know, there's a lot of serotonin in the ventral and orbital frontal cortex. The serotonin transporter is very high there. And when you deplete serotonin there, you get impairments in reversal learning, which you can remediate. And that's one of our models of OCD. Okay. Deplete serotonin in the lateral frontal cortex and look at another form of cognitive flexibility like attentional set shifting. No effect.
Huh.
This is Hannah Clark and Angela Roberts worked with me in, in the early noughties. No effect. Whereas that behavior, this higher order flexibility, seems to be more modulated by noradrenaline and maybe dopamine. Dopamine involved in the learning of a set and the abstraction of a general rule, we think, and noradrenaline involved in switching between rules. So maybe the catecholamines and the indolamines have separate roles in different aspects of cognitive flexibility mediated by different circuits in the frontal cortex. That's the closest I can come to some kind of...
There is some, the density of, say, serotonin receptors on the dendrites of glutamatergic neurons may differ between cortical regions. And I was reading something about layer 4 pyramidal neurons in the prefrontal cortex having very high levels of the serotonin receptor that these psychedelic drugs are thought to.
5-HT2As.
Yeah, 5-HT2A. So you could imagine that you have many different glutamatergic neurons that are innervated by serotonergic neurons, but some of them are relatively more affected because there's more synapses or there's more receptors to synapses or something.
Sounds plausible. There's classical work, of course, by P.M. Magistretti and Floyd Bloom and others, where they looked in the visual cortex and they showed that, as I remember it, serotonin selectively affects layer 4, which is the thalamic input, which of course, and 2A receptors there is going to affect visual receptors. And so clearly you're going to get hallucinations out of that. Whereas noradrenaline fibers tended to innervate more the deeper layers, 5 and 6. So, so there are clear neurobiological differences in laminar innervation of these systems. And you can kind of speculate a bit about what that means functionally, but, we still haven't really nailed that down yet. And also, when are these systems normally active? That's the big question, isn't it? Is it under stress, different levels of arousal? What is it about, the differences in activity in these systems under these conditions? I guess we know most about dopamine, but acetylcholine is clearly very much involved in sharpening signals.
Yeah, and attention.
And attention. That's, that would be our own evidence as well, but many others, I think, would agree.
And that's why, nicotine.
Yeah, nicotine.
Right, makes you more alert and attentive.
Martin Sartre, of course, has quite a nice story on acetylcholine as well, which I think agrees with ours. So that's the really
what? Okay. Go ahead.
Yeah. So acetylcholine is obviously involved in boosting signal to noise ratios, as we and others have shown. Martin Sartre agrees with that, I think. But the key question is, what are these systems doing, naturally speaking, during different states of the world? For example, stress, reward, maybe different levels of arousal or wakefulness. That's the key question to work out, because you may need different executive functions performing under different levels of arousal. For example, if you're in a boring lecture, you're drowsing, you're falling off to sleep, you may need to have a different boost to maintain your concentration and your working memory processing of the lecture than if you're very excited, for example. So that's the kind of consideration we need to have.
Yeah, a better understanding of these mechanisms, may lead to new ways of enhancing, say, cognitive performance and productivity.
Yeah, I mean, obviously cognitive enhancing drugs are interesting.
Yeah.
We've worked with some of those, like modafinil, which does improve certain aspects of cognitive performance, even in healthy, normal subjects, as it were. But it can have some surprising side effects. So I love this, talking about chess earlier.
Yeah.
I love this study that came out in one of the European neuropsychopharm journals. I think it was a study done in Germany of chess players playing against a computer, and they were treated with modafinil.
Ah,
Modafinil improved performance of skilled chess players.
ah.
I think that the less skilled ones improve more than the highly skilled ones. But anyway,
So I should be taking modafinil so I can
maybe,
compete with you.
maybe, but there was a downside to it. The downside is that they lost more on time.
Oh, interesting.
Yeah, it
So
is
if
interesting
there's a more of a time restriction,
that they lost more on time for some reason. So we found, on the Cantab Tower of London task, which is a planning task, that modafinil in healthy volunteers improved performance in terms of accuracy, but it made them slower. And we think what it did is reduce their impulsivity. So it acted a bit like Ritalin, as in ADHD. So it stopped you from responding impulsively and getting it wrong. So you just spent a little more time thinking about it and you got it right. But the downside of that is you lost more often on time.
Yeah. Yeah, I've been playing like really short games where each of us have 10 minutes. And this is kind of a tricky business, right? And early
Yeah.
in the game, how much time do you spend, and are you going to have to rush towards the end?
Well, the secret in chess is to have your openings very well prepared
Yeah,
and then spend all your time thinking at the critical junctures,
that's
which
right.
are about 10 moves in,
Yeah. Yeah.
and then the end game, if you're well read, the end game should be easy.
Yeah.
So you've got to allocate your time efficiently.
Yeah. Yeah. That's right. Okay. Well, we had a great talk today, and
Really enjoyed it, Mark, and look forward to further conversations in the future.
yeah, likewise. Have a good rest of the summer in the UK.
Same to you. Goodbye.
Goodbye.